Hi, Jeff. I don't know who all is here for Berkshire Green Drinks, but Berkshire Green Drinks is happening at the moment. Thank you all for joining us. This event is organized by Berkshire Environmental Action Team, also known as BEAT. My name is Chelsea Simmons. I'm the programs director for BEAT. Sorry, have to remember everything. There is a sign-in sheet floating around. If you haven't signed in, please find that sign-in sheet and sign in. That just helps us keep track of who's attending our events and how many people are attending. Thank you to Hot Plate for co-sponsoring this month's Berkshire Green Drinks and just hosting us. Yeah. And I just want to let you know about next month's Green Drinks. It's going to take place on Wednesday, April 9th at the Racebrook Lodge in Sheffield. It will be hybrid, and Ruben Perilla from NOFA Mass, which is Northeast Organic Farming Association, will be talking about the relationship between glyphosate and soil health. And you can learn more about that on our website and register through our website as well. For everyone on Zoom, if you could please stay muted during the presentation. You're welcome to add comments and questions to the chat while Jessica's talking, and then we'll have a Q&A afterward. And I think the only other thing, so toward the end of Jessica's presentation, she will talk just a little bit about how people can get involved and volunteer to help with the research she's doing. So if you're interested in helping with that, we do have a sign-up sheet specifically for that over here. And then for everyone on Zoom, if you email team@thebeatnews.org, let us know you're interested, and we'll make sure you get those emails. And I think that's everything. You can't hear me?
I have no idea what you've been saying.
Can people hear me? Some people are giving me okay. Well,
Good luck.
Good luck. I'm going to hand it over to Jessica Duffy, our speaker this evening.
I didn't realize there wasn't going to be a mic.
Oh,
So
if you can just.
All right. So that's actually for the people who are watching online. And so I'll do my best. I'm not a big speaker. I did not practice this to be speaking for a big crowd, like without a mic. So I guess I want to say thank you to everyone who came and to learn a little bit more about what I consider to be two really special little butterflies. I want to start with a disclaimer. I guess everybody starts with a disclaimer. Even though I did a master's thesis in the area that I'm going to be talking about tonight, it was a long time ago that I was officially working in the system, not just as a citizen scientist. And so there's probably been other work in this system that I am not aware of. I did try and do a lit search in preparation for this talk, so I just want to put it out there that there may be elements that I'm missing and that aren't going to be included in the talk. So first I'm going to talk about each of the native paired butterflies. There are two. And then they each have individual life strategies and unique challenges that are, and then when we go through that, we're going to talk about garlic mustard and the role it's playing and the pressures that it's also applying to these two populations. So I wanted to start with just the little story about how my project came together. So a million years ago, in the 1990s, I was working on my master's thesis in the lab of Dr. Roy Van Dries at UMass Amherst. And we were looking at 3 paired white butterflies. So that's in the genus Pieris, 1 invasive, and the 2 natives. And then what we were really looking at was that when the invasive species, in a system of biological control, they were releasing parasitoids to try and control the invasive species. parasitoids that may have also have come in with the invasive species. And so my advisor, who was an expert in biological control, was looking at what possibility that these organisms could be having possibly on our native Pieris. So after and I'm going to, and we're going to go into, yeah?
What is a parasitoid?
I'm going to get there. And you're going to hear the word a few times before I get there, but I'm going to explain directly what a parasitoid is.
I have to tell you something, you have to get into your uncomfortably loud voice.
All right.
Really hard to hear.
Okay, it is hard. Okay, so I'm going to yell. So after I graduated.
There's no mic, so she's yelling already.
So
If we all just
I'll try and yell a little louder. I wasn't kind of prepared for that. So after I graduated, I worked for the Department of Agriculture in a couple different roles, and I ended up drifting away from that area of work. We actually ended up living in the city for about 20 years. A nice area, not the right habitat for me. And a lot of what I had learned in graduate school and the experience I accumulated really kind of slipped back to the dark corners of my mind. And then the pandemic hit. And my husband lost his job, we lost our apartment, and my job became remote. We could go anywhere. And we landed in the Berkshires. And there's not a single day that I wake up that I am not blown away that I get to live here. So, but here's the funny thing. So when we were looking at the house that we ended up buying, we were walking the yard with the realtor, and one of my kids was like, "Hey, there's a little white butterfly here without spots on its wings." I stopped dead in my conversation with the realtor, and I went to see. I bet you he thought I must have been kind of crazy. But indeed, in the backyard, there was indeed a little white butterfly without spots on its wings. And at this point, I could hardly even remember why this was so significant in my mind, other than I knew it was probably one of the butterflies I had worked on in school a million years ago, and that possibly an endangered species. But it took a while for it to all kind of land in my mind. But then, whoops, I got a dog. And the dog is kind of a key to this, because if I did not get, this is Izzy, by the way. If I didn't get Izzy up the mountain every single day, we're about a mile from October Mountain, she'd drive me to drink, right? I mean, I work from home. So there's no way I was going to get any work done with a fidgety, whiny husky tip-tapping all over the house. So while I was up on the mountain, I found myself kind of compulsively picking garlic mustard. I knew it was an invasive species, but it didn't have anything to do with any of the work I did in school. It was just kind of, it's invasive. I pulled it, satisfying to pull, and your good deed for the day. But then I started to cue in on the first spring host plant of the butterflies that I was working on. And I was surprised that I even still remembered it. And then there were the butterflies, like right in front of me, just flying around. I mean, it was like seeing a celebrity. And then, it's like all the connections kind of said, like, bam, came back. And now here I am. And so these are the stars of the show. So I was looking at these. So the first butterfly here on the left is the cabbage butterfly, Pieris rapae. And as you can see, this butterfly has spots on his wings. So I know it's a male because it has one spot on each forewing. Females have 2 spots on each forewings. And this is an invasive species. This butterfly invaded through Canada around 1860. And by 1869, it had reached Boston. A lot of people call this a moth, the cabbage moth, if that's a more familiar name. Gardeners might know it as moth, but it's definitely a butterfly. And you might recognize the little green caterpillars on your cabbage and kale as cabbage worm. That's this guy. So the next one, this is the West Virginia white butterfly, Pieris virginiensis. Now this butterfly is rare, but it's not necessarily threatened that we know of, at least not yet. The reason it's rare is because it has a univoltine life cycle, which means it has a single flight. So it comes out really early in the spring. It overwinters as pupae. It comes out really early, between late April, early May. It'll lay its eggs on the host plant, which I'm going to talk about in a minute, which is called toothwort. They develop through, become pupae, and that's it. And they're only flying in wooded habitats because that's where the host plant is. You have maybe a 2 or 3 week window where you may run into this butterfly in the woods if you happen to be in woods where its host plant happens to be. So, but as far as we know, its populations are still okay, but I think garlic mustard's going to play a big role in the future health of this butterfly's populations. And then the last one is the veined white butterfly. I think its more common name is the mustard white. I call it the veined white butterfly because it has these gorgeous outside, like outlined wing veins, and that's in its first generation. It has a second and a third generation in July and August, where these wing veins are more shaded out, or you might not see them at all. Now, before cabbage butterfly invaded, this was your garden pest. It was a fairly ubiquitous butterfly, and this was could have been doing damage to your agriculture. Again, it's for brassica, your kale, cabbage, cauliflower, broccoli. That's what it's eating. But now it's a threatened species, and it's almost eliminated from its southern range. Now this is toothwort, two-leaf toothwort. And it's found up on October Mountain and other places that tend to be higher elevations, moist, cool. And this particular plant is the critical host plant for our native Pieris, for both of them. It's the only host plant for the West Virginia white, and it is a critical first host plant for that first generation of the veined white butterfly. It's one of the earliest forest crucifers to come up in spring, particularly in those moist, wooded, riparian habitats. It's a spring ephemeral. You're going to find it in the same places where you're going to find trillium and trout lily, things like that. When the canopy starts to close in, it starts to senesce. All right, so just a little bit more about the West Virginia white here, and then I'm going to come back around to it towards the end in relation to garlic mustard. So if you look at the range map for the state, all the known populations are right here in the western part of the state, and they coincide directly with the presence of toothwort. Some of the early work I did in my program was we surveyed road stream intersections all across western Mass and central Massachusetts in forested habitat to see if there was toothwort that didn't coincide with the population of butterflies. And there was no toothwort in the central part of the state, and where there are no butterflies. And where there's lots of toothwort, you're going to find the West Virginia white, and sometimes the veined white. More broadly, you can see the West Virginia white in the northeastern United States, usually at those higher elevations, like along the Appalachian range, anywhere where it's cooler, moister, forested, and there's toothwort. So the West Virginia white, with that univoltine life cycle that happens so early in the system, or so early in the season, has built itself this like really elegant little niche, right? So it comes out and it's done before a lot of those mortality agents are really getting going. So they're not going to have the same pressures from predation and disease and that word parasitoids, and I'm getting to that, either. So it's like this little niche, but because it's so specific, it's a precarious little niche. All right, now my baby here, this one. This is, so a lot of the work that I was doing in school originally was more on this butterfly than the West Virginia white. Because the West Virginia white has only that one generation early in the season, you can't maintain a colony. You can't keep it going and do experiments with it. It's here and then it's gone. But this is actually the one that's more critically threatened in the system I'm talking about. So this one also, their flight happens really early, primarily in that wooded riparian habitat. Last spring, I actually saw one of these fluttering around near the forest floor at the end of April, and it was chilly, especially for an insect. And like the West Virginia white, this first generation is tied to toothwort. However, in later generations, in the second and third generation, flight and generation, this butterfly branches out, and it can use your garden brassicae and wild mustards and cresses, so it has a greater host plant breadth than the West Virginia white. But that second and third brood tend to be really heavily impacted by pesticide use in agricultural crops, in brassica crops specifically, as well as disease and parasitoids. I'm almost there, I swear. So, as I mentioned, it's a threatened species. As of an article that was published on the Massachusetts website, mass.gov, this butterfly is only found in 5 towns in Massachusetts, right here in central Berkshire County, and my house. However, it's not necessarily threatened in other parts of its range, which runs across southern Canada and the northern United States. It has a fairly broad distribution, especially if you compare it to that of the West Virginia white. It's along its southern range of the southern range of its former distribution that its population's declined. So in Massachusetts, it's listed as threatened. This graphic calls it imperiled. It's vulnerable in New Hampshire and in Ohio, and considered critically imperiled in Indiana. However, after the mid-1800s, the experts of that time noted a really rapid and dramatic decline of the veined white butterfly that they correlated directly with invasion of the cabbage butterfly. So one of those experts, writing in 1889, was S.H. Scudder, who was a Harvard entomologist. And so 1889 that's only 20 years after the cabbage butterfly hit Boston, that was at least officially documented. And Scudder wrote, quote, "The invader has nearly exterminated the indigenous species. I recollect once seeing the college yard," and he's talking about Harvard University, in Cambridge. I think it was about 1857, fairly swarming with P. oleracea, and that's the veined white. It is now never found, to my knowledge, anywhere in the region about Boston. Even in the recesses of the White Mountains, I have never in recent years seen it. So common is P. rapae." End quote. It was also noted generally at the time that the veined white had come to be more associated with wooded habitats. So Scudder went on to say, quote, "In keeping with this, it seems to be a far more timid species than P. rapae. It is if, in its partial supplanting by rapae, it felt that every man's hand was against it." I love that quote. But so at the time, the scientists of the time thought that the decline of the veined white was related to more aggressive behavior of the invasive species, forcing the veined white into more wooded habitats. However, there's been research, most notably by Dr. Frances Chu of Tufts University in the 80s and 90s. She was a huge player in this system here. She did amazing work. But she found that where these two species are still found together farther north, they occupy the same areas. So then the question became, how did the cabbage butterfly push out the veined white butterfly so dramatically after it invaded? It's not like there's a shortage of brassica crops and wild crucifers. So there had to be something that wasn't necessarily related to resources. So my advisor, Dr. Van Dries, he's an expert in biological control, began looking at potential non-target impacts of parasitoids that were released to control the cabbage butterfly on the veined white butterfly. All right, here we go. So what's a parasitoid? So parasitoids are almost always wasps. So they're in that same family with bees and ants, so they're wasps. Occasionally, some parasitoids can be flies, and parasitoids are different than parasites in that they use their host, they lay their eggs in or on their host, which will then die from the infestation. So it's like Alien, the movie, right? The larvae grow inside the host, feeding until it bursts out to pupate. And that's as opposed to parasites, whose strategy is more successful, where they can progress through multiple generations with the host alive. Fleas and lice aren't going to get very far if their host is dead. So parasitoids are often used in biological control of some invasive species. The aim of biological control is to take natural enemies from the areas of origin of an invasive species and then introduce them as an agent here in the new area, right? Because in their native habitats, the invasives, whether they are plants or insects, have evolved with their natural enemies in this whole system of checks and balances. So for example, in the UK and Europe, garlic mustard is everywhere, but it doesn't take over like it does here. It's maybe the equivalent of Queen Anne's lace or dandelions. It's a weed, but I got online and there are plant nurseries in the UK that will sell you garlic mustard to plant along your hedgerows to try and attract native butterflies. So, now this picture was taken in my garden 2 years ago. So the two parasitoids we were particularly interested in, and unfortunately they don't have common names, which is kind of a bummer, but the first one is Cotesia glomerata, and what you're seeing here is a mass of cocoons that have just exited this caterpillar. So they're a larvae, they came out, they wove their cocoons. So you can think of glomerata, glom of eggs, a glom of larvae. And then, so they were feeding inside. So I guess I should say, these wasps are teeny. They're teeny, teeny, tiny. You would really not see them with your naked eye unless you were really, really close. This caterpillar was parasitized probably not long after it hatched from its egg. So in addition to being released to control cabbage butterfly, these wasps almost certainly invaded along with the cabbage butterfly. So when that shipment of cabbage or whatever came in from England or the UK or it had probably already been infested by cabbage butterfly larvae, and some of those larvae had probably already been parasitized before they even left. So the second one, Cotesia rubecula, was released later in the 80s, I believe in the 80s, under much more strict biological control testing protocols. I actually had a conversation with Dr. Van Drijs last fall when I started really getting into kind of this project. And he suggested that Cotesia rubecula may be outcompeting glomerata, which would be really good news. And that was from research that was done more recently by one of his students actually here in Canoe Meadows. So I'm actually hoping to do some work in Canoe Meadows as well. So I'm going to go through, I have a couple data slides I'm going to go through. I want to explain I don't have pictures because so much of this work was done so long ago, but just to kind of give you an idea of how we ran the experiments, because these wasps are so tiny, right? We're not just going to catch them. So we needed to bait them. So basically, we maintained colonies. We had a colony of cabbage butterfly, colony of veined white butterfly, and colonies of each of the parasitoids. And then for the field experiments, we would take potted collared plants, so like, in a basic 8-inch kind of nursery pot, and we'd put some veined white caterpillars on one collared plant and cabbage butterfly caterpillars on another collared plant, and we would take them and we would put them in agricultural fields of brassica crops, as well as meadow and forest habitat where the veined white butterfly is present and absent in northern Vermont and western Massachusetts. So we would place them in the field like maybe like a meter apart and just see who found them. Those are called trap hosts. So we're kind of trap hosting sentinel larvae. And then after 3 or 4 days, we would bring them back to the lab, find any surviving caterpillars, dissect them, and find out if they had been parasitized and by whom. So I also want to say, I guess one of those disclaimers, that I did cherry pick the data a little bit for the purposes of this talk. So I picked the most straightforward data, but just know that the trends that I'm going to talk about held true through all of our experiments for the whole 3 years I was working in the system. So the data is representative of the system overall. So in this case, this is for, we started with laboratory comparisons. And right away, you can see when Glomerata was presented with cabbage white and veined white larvae, they were picking the veined white larvae 2 to 1 over the cabbage white butterfly. So it's already showing a really strong preference for the veined white caterpillars. Rubecula was a little bit more ambiguous in the laboratory tests for some reason. We don't know why. But they did show a preference for the cabbage white overall, though they seemed perfectly happy to parasitize the veined white under laboratory conditions. I'm not really sure exactly what was going on there. But then in the field, Rubecula, good little bug that it was kind of at lower levels, never parasitized a veined white larvae in our field experiments. And that held true the whole time. Not once did we get a field larva back that had been parasitized by Cotesia rubecula. Glomerata was blowing away the veined whites at a rate of 6 to 1, 5 to 1, 6 to 1, depended on which week we had them out. This almost seemed like they might have hit those by accident. The preference was so strong for the native species. And then, but there was another piece of the, another piece was also the scientists of the time, when they were first seeing the real sharp reductions in the veined white, noticed that they tended to be, if you were going to see them, they were going to be in wooded areas. And so we did in this experiment, just using the cabbage white larvae, because we weren't comparing between the 2 different species, we wanted to see how those parasitoids would forage. And so we got kind of what you expected. More foraging, more parasitoids in agricultural fields. The adjacent meadow, Glomerata was more willing to kind of forage out and look around. But we really never got parasitism in once that transect line hit forested points. Once or twice, I think we got twice parasitism by Glomerata in the forest. And also there's a native wasp. For those who like it's in the family Ichneumonidae and genus Hyposoter. But they hit a couple, just a couple, nothing that was going to affect the population. So it seems to explain what earlier entomologists had observed after the cabbage butterfly invaded, kind of seems a better explanation for why the native species declined so sharply. As the cabbage butterfly expanded its range, so did Glomerata, and was potentially blowing away our native species along the way. What these sets of data don't explain, though, is why the veined white butterfly is still doing okay in northern parts of its range. So this is actually data from some work that was done after I left, but also done in Dr. Van Drijs's lab. So first of all, diapause. Diapause is a state where the insect stops their development at some specific point. Such as in this case, they'll reach pupation, and then they go into diapause. So they start to develop, and then they stop. All those little chemical reactions stop for some reason until the following year. So in the case, and so this is first generation on this middle line here. So that's the generation that came out first early in the spring. They laid their eggs on toothwort. And of the, in Vermont, that first generation, 86% of that first generation went into diapause to the following year. And that's compared to Massachusetts, where it was 63%. And that's actually really good because the parasitism in that second generation, so of the 37% that didn't go into diapause, they became butterflies, they laid eggs and caterpillars. 100% of those caterpillars in Massachusetts were parasitized by Cotesia glomerata. So the paper that reported this data came to the conclusion that the second generation in Massachusetts is unlikely to produce many, if any, surviving pupae for a third generation or the following year. Luckily, they're caching some, and Vermont's caching a lot. So one thing that they noticed was that if the larvae developed on toothwort that was a little bit farther along, they were more, so once it was in a budding stage, they were more likely to go into diapause. Now in Vermont, the toothwort's coming out sooner because the toothwort's responding to day length, but it's cooler. The insects, in order for the insects to come out, they need what's called an accumulation of degree days. It has to be warmer for a certain amount of time before they can come out as butterflies. So there's, I guess the issue is going to be, is that the next one? Right? So climate change is going to play a role as well. So as it gets warmer, those butterflies are going to come out sooner. They're going to be hitting the toothwort sooner at an earlier stage. So it's likely that there'll be a smaller percentage that will go into diapause to be able to mitigate that parasitoid threat. And now enter garlic mustard. These poor little guys, I don't know. So garlic mustard, like toothwort, is an early spring crucifer. It's really aggressive, as I think everyone knows. Nothing eats it, not even deer. The other thing about garlic mustard is that it's one of the few opportunistic species that can really invade understory. Most require disturbed habitats to grow. And this has allowed garlic mustard, and you see it crazy going up on October Mountain, really invading those wooded stream-side habitats that have been the home and refuge of our spring ephemerals, like toothwort and trillium and trout lily, and the butterflies that rely, and how many other creatures rely on that particular ecological system. All right. So in the case of the butterflies, garlic mustard could also be acting kind of like a super brassica to the senses of the butterflies. So like McDonald's, right? It's easy, it's apparent, it's abundant, it's kind of everywhere, and with probably stronger biochemical signals to attract the butterflies. Both toothwort and garlic mustard contain a compound called sinigrin. And sinigrin is thought to play a role in eliciting oviposition behavior in the butterflies. But it also contains those natural defensive phytochemicals that can deter larval feeding. So the females lay their eggs on the garlic mustard, potentially, and that's what I want to look at more this season. But then the females don't develop. This is going to be particularly true, I believe, and again, I'm hoping to get better data this year, for the West Virginia white, because the West Virginia white doesn't have that host plant breadth that the veined white has. So Dr. Chu also, she did an experiment where she was rearing, so it turns out that the veined white can develop on garlic mustard to a certain extent. So in her experiment, and this was back in the 90s, she placed 34 first instar veined white caterpillars on the garlic mustard, 14 of which survived to pupation. And it took them 23 and a half, give or take 3.5 days to get to be able to pupate. Compared to hare's ear mustard, which is also an invasive mustard, but apparently a more suitable host for the veined white, it only took 14 days, give or take a half. So it's a 50% longer time to develop on the garlic mustard. And that longer development time is really significant because the caterpillars are going to be vulnerable to predation, disease, and as we've seen, parasitism for a much longer period of time. So garlic mustard is physically crowding out that, especially toothwort, that key first early host plant for these butterflies. Just physically crowding out. It also is changing the soil chemistry, which is going to affect the whole ecosystem altogether, and potentially acting as an egg sink, especially in regards to the West Virginia white. But here we come back to those little wasps. The garlic mustard could be acting like a biochemical highway, a corridor for those wasps to follow into the woods where they would not have been able to forage before. And I kind of think that emerald ash borer on the mountain might have a role in that, because it's really opening up a lot of the forest canopy is being opened up by all those trees coming down. The garlic mustard is offering like a biochemical highway, and so then the native butterflies may lose their last refuge from those parasitoids, as well as losing their critical host plants. Okay, so this is the access road from the end of New Lenox Road up to the Farnham Reservoir Dam. This is where I've been casually following, specifically the West Virginia white is here. Over the last couple of years, there's plenty of toothwort and a seemingly expanding infestation of garlic mustard. To me, it seemed a lot worse last year than the year before. So what this represents is a nice, finite, and accessible area from which we can survey the West Virginia white as we pull as much garlic mustard as possible to see how this will affect the prevalence of the West Virginia white over time. So I'm trying to create basically a citizen science project, a research project, survey the West Virginia white. Oh, did I do that? That's all right. Over here. So along that corridor, what I need assistance with, so this is kind of the volunteer plug, is I need some volunteers to walk that access road where I've been observing the butterflies. Because I have this day job, unfortunately. So I can't get out and observe the butterflies during those prime flight times at like midday where it's warmer, right? It's a cool time of year. So they're only really getting going in that kind of that 11 to 2 range where it's warmer and they can really go through all their behaviors. So I'm hoping there may be a few people who might like to hike up to Farnham Reservoir on lovely spring days between possibly April 1st and May 31st, so that we can make sure we catch both ends of that flight and grab some data, like how many butterflies are you seeing? Where along the route are the butterflies? How are they interacting with the garlic mustard and the toothwort? Are you seeing egg-laying behavior, nectaring? This would be really, if we see egg-laying behavior, we can possibly take some agricultural fabric and cover those garlic mustard plants, cinch them to protect those larvae from predation and parasitoids, and then see if they can develop through. Somebody may have done this research before, but I haven't come across any of the lit on that myself. Another volunteer opportunity will be a couple of organized garlic mustard pulls with beet on a weekend or two. The Lenox Garden Club helped with this a little bit last year as well. So we'll be bagging the garlic mustard, and Second Chance Compost is going to take the material. Regardless, we will be removing it from this finite habitat location so we can see how and if the West Virginia white population will be affected. I do have one other I'm quick, okay. So I also have another kind of set of experiments, and that's going to involve taking kale plants and placing them out where I know there's veined white butterflies and cabbage butterflies, hopefully, and seeing if I can get those plants to be inhabited. Leave them out long enough for them to have eggs laid on them, for them to develop into caterpillars and get parasitized or not. Cover them with, again, agricultural fabric back at my house and see what I rear out of them. What percentage of veined white butterflies are actually out there? What percentage of cabbage white butterflies? And which parasitoids, if any, are attacking these butterflies here? Again, because we're the last refuge in Massachusetts for the veined white. This might actually be a good undergrad project if somebody who needs like an undergraduate project. Should be some interesting data and a fun little project. And yeah, so I think that's it. I'm ready for any questions. I'm sorry for anybody who might have had trouble hearing me. Yes?
How in the world does one recognize egg-laying behavior?
Sorry. I didn't mention this. Can you repeat the question for the people on Zoom? Or we can walk around, but it would be quicker.
So she wants to know how you would recognize egg-laying behavior. It's actually kind of obvious when you see it. So the butterfly will be landing on a plant, and it's actually going to like tuck its little bum underneath the leaf, and then there'll be an egg. Like if you go up after, you'll be able to see it's like a little spindle on the bottom of the leaf. But it's really specific behavior that once you see it, you'll know. Yeah. Any other questions? Yes.
So the data you collected, just so I have the timeline right, that's when you were in college or that's when you were studying and you're just now sharing it with us?
Yes.
And
Well,
you're comparing it to what you're seeing in your backyard in October Mountain?
basically, yeah, it's kind of like, so I went to school and then I came here and they were all there and I was like, and I just kind of got back into it. And so I want to get new data and possibly better data. I don't know exactly what's going to happen, but there's a lot of questions and there's a lot of fun ways to figure it out. So, yes, yeah.
If we were to walk up October Mountain without you, and we pick garlic mustard, what is the best way that we can make it go away, dispose of it? Repeat the question.
Okay. So somebody's wondering, like, so if you go up and you're picking garlic mustard, what can you do with it if you don't have, second chance compost to track it to? That's a really good question. If it's not a ton, a lot of people will crush it, they'll leave it in the road to get crushed. I think if it gets to flower though, you're still gonna get the seeds going. Burning is an option. So, I mean, I've always just bagged it. Technically, you're not supposed to put it in your trash. But, again, if everyone's just doing a little bit, then it makes a big difference. So, wait, let me go over here first. Sorry. What's the question?
What about solarizing like plastic bags?
Oh, that would be a lot of plastic. So it would be a lot of plastic. I mean, the whole mountain is infested. So the question was whether you could solarize it with covering in plastic to kill it that way.
Well,
I mean,
pull it. No,
oh, pull it?
dispose it.
Oh, pull it and then solarize it.
Yeah,
It's something to consider. I'm not sure, I guess, where. I mean, you could do it, at your home. Like, if you're going to do it at your home, then.
well, we're doing it in a public park in Monterey School Village.
Oh, okay. Yeah.
And we're going to be doing garlic mustard pool very soon. What should we do with all the garlic mustard we pull?
Okay. So small park. I mean, that's a possibility. As long as you pull it before it's really going into flower, you got to get it before it goes. I mean, even when it's flowering, it can still set seeds, but as early as you can get it, and if you could try that, I don't know for sure, but if it's not too much.
Compost it otherwise?
So composting, it gets to a high enough temperature that it will kill the plants and the seeds. He swears that John Petroff, who runs Second Chance Compost.
Industrial composting,
Yeah.
not backyard composting.
Yeah. But at the same time, for the purposes of the study, we're removing it. The whole point is to just remove it and see if it makes a difference to the veined white, to the West Virginia white population. So it's kind of a finite experiment. It's not it's not trying to be a way to completely eliminate garlic mustard. We're really just trying to eliminate garlic mustard in this finite area to see how it's going to change, if it makes a difference. Sorry, yes?
Yeah, but can you, because it looks like there might be some individual who will pull garlic mustard on their own.
Yeah.
Can you describe how to do that? Because there is a right way and kind of a wrong way.
Okay, I mean, you want to get the roots. Garlic mustard is fairly satisfying to pull. As long as you get the roots. The key is, once it goes to seed, and you can see the little seed pods on it, at that point, pulling it, you might just end up dispersing it more. So,
what you should be aware is that the root actually makes like a curve, a turn, like this. And so if you just innocently pull it out straight like that, you'll break, the plant from the root. The root will still stay there. So you need to kind of like really dig in and get to that curve and then gently pull it out.
well, and also too, there is a seed bank. So it's something that's not going to just happen in a single year. Right. Like we really have to go, like for it to work, we'd have to go back for several years. I anticipate working on the project, for 5+ years. I'll be pulling garlic mustard till the day I die. So again, it's kind of a long-term kind of, I think there were some questions over here. It'd be a long-term commitment.
I also am going to walk around with the speaker and have
Okay, sorry.
right into the speaker.
Okay. So this
Wherever you wanna
come back. So
get to everyone
yeah,
eventually.
yeah.
Hi. I was wondering, so have rubecula or glomerata were they both introduced, I guess, on accident, or are they a tactic to repel the moths or butterflies?
So both.
Yep.
So the question was whether these came in by accident or whether they were deliberately introduced. So in the case of glomerata, they did come in. We're almost certain that the loss of the veined white butterfly starting back in the 1800s started with the invasion of cabbage butterfly with glomerata. But then in the early 1900s, in those early glory days of biocontrol, they did release it as well to control cabbage butterfly in biological control. Rubecula was only released specifically and deliberately to control cabbage butterfly. It was not an accidental introduction.
And you are seeing, you think that's trending towards rubecula replacing the glomerata as well.
There's some data, and that was just from a personal communication with Dr. Van Drijs last fall, that there is the possibility that where rubecula has been released, it may be able to outcompete glomerata, which would be very helpful.
Yeah. Nice.
So.
Thank you.
And I think there was someone, yeah?
So last spring, I went on a ephemeral walk with the Berkshire Natural Resources Council up in Hinsdale on the Old Mill Trail, filled with garlic mustard. And the woman who was leading the walk said they had all these volunteers pulling out all the garlic mustard, and they really did a good job of clearing it out. But unless it's replaced with another plant, the next year it all came back.
Well, it's going to it will. So so there's the seed bank,
Yeah.
and you're really going to take a few years. It's not going to just all go away at once for sure. So we kind of have to wait to pull out and get through that seed bank. And it might not be a permanent thing, but when we can get it down, then we're not dealing with those masses. I don't think there's any way we're going to really eliminate garlic mustard. The purpose of the experiment that I want to do is really just to eliminate in a finite area so that we can see how it affects a particular population of butterflies.
I wonder if once you remove the garlic mustard, if you plant something else there, it'll prevent the garlic mustard from re-emerging.
Yeah, I don't know. I don't think, and I don't have the resources
for that.
I mean, it's something I can maybe kind of keep in the back of my
The
mind.
Berkshire Natural, they're looking at that, though, I think, the
Okay, yeah.
Berkshire Natural Resources Council.
Yeah, cool. There was somebody over here. Was there somebody over here? I'm sorry. So that's for the virtual, for the people who are watching virtually.
Well, I appreciate your talk, and I can't wait for the experimentation phase, for sure. I guess I have what it sounds like kind of an unserious question, but I mean it seriously. Do you feel like your dog gets in the way of your observation?
It's hard to pull when you're tethered. Like being tethered, so, definitely slows me down for sure. So, but then I go back, and my son has helped a lot. We go back sometimes, like after work, and we'll do some more pulling. And then hopefully if we can get a couple big pulls with a lot of people, then I think that we'll be able to get a good dig into it, so.
Great. Thank you.
Oh, I'm so sorry. Hi.
Hello. Is it easy to buy toothwort to plant?
I have no idea. She was wondering if it's possible to buy toothwort to plant, and I don't know if that's true. I don't know if you can.
We'll figure it out.
Yeah, no, that's another good thing to kind of put in the back of my head.
Good spot to look is Alliaria Native Nursery down in near Great Barrington. I don't know exactly what it's on. It's all for I know it's everything. They sometimes have these these forced understory herbaceous plants.
Oh.
And it's just that's one spot to look.
Okay.
We have a question on Zoom.
Oh, okay. Should I go over?
So I believe the question was, are there any insects or mammals that rely on garlic mustard around here? Who relies on it?
Nobody. No, they really don't. No, it's true. Nothing eats it. Nothing eats it. They've done a lot of experiments on that, and really nothing utilizes it. So.
I should point out that humans are very welcome to eat garlic mustard.
Right.
It has good nutrients, and some people find it tasty. Oh, you can make pesto.
Yeah, like the pesto, and they're working on it, like trying to develop a market for it, but.
So one more question, or you can just wrap it up.
You could probably eat the flowers. You can eat the flowers are so tiny.
There is one person over here. So
One more? Oh,
So
sorry.
we only have time for one more question.
This isn't a question, it's just a statement. Yes, you can buy toothwort to plant, according to these smartphone device
Okay.
things.
You can buy toothwort.
Lovely. As seeds or as plants?
As plants.
Okay. Okay. And we might have to do that if they.
Yeah, the fact that they're not expensive either, so the more
Okay.
you buy, I guess.
You can get any.
That's good to know. Good
All
to
right.
know. Thank you.
Thank you, everyone. Thank you so much to Jessica for coming out tonight and presenting. Hot Plate will have a DJ after this if anyone wants to stick around. Again, also we have just a sign-up sheet if you're interested in volunteering and helping Jessica and any of the research and work she's doing. And if you didn't sign in, please do so. It really helps us beat who organizes this event. And yeah, thank you all.
Garlic mustard.